Imagine a great white shark. Its eyes are covered, but it's still somehow zeroing in on its prey. It's not using sight in the way we think of it. No, it's literally detecting these tiny electrical fields from living things hidden in the water.
Yeah, or think about a bat, flying in pitch blackness. It's sending out these high-frequency clicks and using the echoes to build this incredibly detailed, 3D map in its head of everything around it. It's just a completely different way of experiencing reality.
Totally different.
Leaf.
Welcome to the Deep Dive.
Yeah.
Today we are taking a really deep dive into the fascinating world of animal sensory physiology.
Right.
We're using your source, Animal Physiology, From Genes to Organisms, 2nd edition, as our guide.
Right.
We're going to try and unpack how all sorts of creatures, from tiny bacteria up to the biggest whales, gather information not just about the world outside, but their own internal states too. So our goal here is to trace how sensation works, starting right down at the molecular level, the cells, and then building up to see how whole systems, even ecosystems, are shaped by these amazing sensory abilities.
Yeah, and we'll look at everything from simple reflexes to, really complex perceptions. We'll dig into some wild experiments, how genes connect to what these senses actually do, and the whole evolutionary story behind them.
Okay.
And what's so fundamental, really, is how essential sensing is to life itself. I mean, it's not just about seeing or hearing like we do. It's how an animal keeps its internal balance. Homeostasis finds food, avoids becoming food,
Right.
and even handles complex social stuff. It really makes you wonder, how do these really simple cellular bits scale up to become these incredibly diverse and powerful senses we see everywhere?
Yeah, that's the big question. Okay, so let's start right at the bottom. Before we had complex brains, how did life even begin to sense anything?
Well, the story really starts with the absolute basics. Cellular things like ion channels and receptor proteins, these are incredibly ancient. You find them even in prokaryotes, single-celled life.
Wow.
Think of them as the very first detectors. They're specialized to do something called transduction.
Transvection. Okay.
Which basically just means converting some kind of external stimulus, like maybe light, maybe a chemical, into an electrical signal the cell can actually understand and respond to.
Got it. So a physical nudge or a chemical signal gets turned into electrical energy. And that's where the evolution of these gated channels comes in, right? That sounds super interesting.
Exactly. The earliest ones are probably mechanically gated channels. They're sensitive to physical stretch. Maybe they first evolved to sense osmotic stress, like if a cell swelled up too much, the channel would open, triggering some kind of response.
Right.
Then you get chemically gated channels, also ancient, for sensing chemicals. Think about chemotaxis bacteria swimming towards, say, sugar molecules in a cow's stomach.
Right, following the food.
Precisely. And connecting this up, chemoreception is actually kind of surprisingly central to how multicellularity even evolved. You see it in things like slime molds.
Slime molds? Really?
Yeah. When these single amoebas start starving, they release a chemical signal. It's cyclic AMP, or KMP. This acts like a beacon, a chemoattractant, drawing other amoebas together until they aggregate and form this multicellular slug structure. It's amazing. And that basic chemical signaling trick, animals kept it and adapted it for all sorts of sensing.
That's wild. And even voltage-gated channels, the ones that are absolutely critical for our nerves.
Yeah.
They're ancient too.
They are. Their original job in bacteria like E. coli isn't totally clear, but we find them in yeast, in plants. Think the Venus flytrap snapping shut. That involves voltage-gated channels. But here's a really key point for animal evolution. The specific sodium ion channels that allow for really fast, long-distance electrical signals, those are almost entirely an animal thing.
Okay. That's a big branching point.
Definitely. And we even see thermally gated channels in some ancient protists, like paramecium. It's incredible. You can almost think of a paramecium as just a single, free-swimming sensory cell. It reacts to heat, touch, chemicals, light, ions.
Wow.
It really shows that a lot of the basic toolkit for sensory neurons was already there way before complex nervous systems came along.
So taking all that in, how do scientists actually categorize all these different sensors in animals?
Well, generally they classify them in two main ways. First, by their modalities, basically, what kind of stimulus they react to.
Okay, like light or sound.
Exactly. So you have mechanoreceptors for touch, pressure, sound, balance, chemoreceptors for smell, taste, internal stuff like blood oxygen, thermoreceptors for heat and cold, photoreceptors for light, And then the more unusual one,
Or the stranger ones.
electroreceptors for electric fields, magnetoreceptors for magnetic fields, and you also have nociceptors, which are pain receptors, often detecting extreme mechanical or chemical signals that mean tissue damage.
Got it. Modalities. What's the other way?
The other way is by their roles, what kind of information they're gathering for the animal.
Okay.
So interoceptors monitor the internal environment, think blood pressure, body fluid composition, crucial for homeostasis. Proprioceptors tell the animal about its own body's motion and position, like where its limbs are. And then exteroreceptors are what we usually think of as the senses: vision, hearing, taste, smell, touch, temperature, detecting things outside the body.
So modalities and roles. That framework helps make sense of it all.
It does. It shows how these fundamental cellular mechanisms get adapted for totally different jobs, from keeping the inside stable to navigating the big wide world.
Which brings us to a really fundamental point, doesn't it? Is what we perceive or what any animal perceives actually reality?
Yeah, and the answer is a pretty clear no. Our senses, any animal senses, are incredibly limited. They don't just passively receive information, they actively filter it. They enhance certain aspects.
Right, like our own eyes sharpen edges and make motion stand out.
Exactly. And our brains go even further. They fill in missing information. Sometimes they even create illusions. Think of the great bowerbird building its little courtyard.
Oh yeah, the perspective trick.
Right. Arranging stones to make itself look bigger to impress a female. That's not a flaw in its perception. It's an evolutionary adaptation. It's about extracting the most useful information from a messy world efficiently.
It makes sense.
Yep.
But it also opens up these tricky questions, doesn't it? Like, do fish feel pain?
That's a classic, really difficult one. Studies show trout doing complex things when injected with irritants, stuff that's not just a simple reflex, but they lack certain brain areas, like the neocortex, that we associate with pain in mammals.
So the jury's still out?
Pretty much. It just highlights how incredibly hard it is to really get inside the head, the subjective experience of another species.
Definitely. Okay, let's get into the mechanics then. How does a stimulus, light or pressure or whatever, actually become a signal the brain can use?
So every receptor type has what's called an adequate stimulus. That's just the kind of energy it's evolved to be most sensitive to.
Like light for photoreceptors.
Exactly. And this ties into Mueller's doctrine of specific nerve energies. Basically, it says the sensation you experience depends on which receptor got stimulated, not what stimulated it.
So that's why you see stars if you get hit in the eye. The pressure hits the photoreceptors and the brain just says, okay, signal from the eye pathway. Must be light.
Precisely. Now, once that receptor is stimulated by its adequate stimulus, its permeability changes, creating a receptor potential. This is a graded potential.
Meaning its size matches the stimulus strength.
Right. Bigger stimulus, bigger receptor potential. Now, in some sensory neurons, this potential can directly trigger an action potential further down the nerve fiber. In others, like in your retina, the receptor cell releases a chemical messenger, a neurotransmitter, which then triggers the next neuron in the chain to fire an action potential. It's like a little handoff.
Okay. And a stronger stimulus. It doesn't make a bigger action potential, does it?
Yeah.
Because they're all or none.
Correct. The action potentials are always the same size. So how does the brain know the stimulus is stronger? Two ways. Frequency code, the neuron fires action potentials more rapidly, and population code, a stronger stimulus activates more individual receptor.
More signals coming faster. Got it. Now, what about adaptation? That seems really important, like how you stop noticing the feeling of your clothes after a while.
Absolutely critical. Some receptors just sort of get used to a constant stimulus and reduce their firing rate. We call receptors that adapt slowly or not at all tonic receptors. Think about muscle stretch sensors. You need constant information about your posture.
Right, you don't want those adapting.
Definitely not. Then you have phasic receptors, which adapt really quickly. They're great at signaling changes. Your touch receptors for clothing are mostly phasic. You notice putting clothes on, but then the sensation fades.
Until you take them off, then you notice the change again, the off response.
Exactly. And this is incredibly useful. Your brain has limited bandwidth, right? Ignoring constant, unchanging, unimportant stuff prevents sensory overload. It lets you focus on what's new or different.
Makes sense. Predators probably use this too, right? Stalking slowly.
For sure. A lioness moving very slowly might not trigger a strong response until that final sudden pounce. Adaptation can be a matter of life and death.
So how does this adaptation actually work? What are the mechanisms?
Well, some adaptation is intrinsic, happening right at the receptor. Take the Pacinian corpuscle, that pressure receptor in the skin. It has these onion-like layers that mechanically absorb sustained pressure. Plus, its sodium channels tend to inactivate quickly.
Okay, so physical structure and channel behavior.
Right. Or in your nose, for small receptors, calcium coming into the cell can trigger a process that closes the ion channels. That's why a smell seems to fade unless it's something potentially dangerous, like smoke or rot. Then you have non-adapting cells to keep you alert.
Interesting. Is their adaptation controlled by the brain, too?
Yes, that's extrinsic adaptation. Think about hearing. Your brain can actively filter out constant background noise, like an air conditioner hum. It uses inhibitory signals, but you can consciously choose to listen to that hum again instantly.
Ah, so the brain has override control.
Exactly. It allows you to dynamically shift your focus in a noisy environment.
Okay. And once these signals are generated, how does the brain know what they are and where they came from? You mentioned labeled lines.
Right. Every sensory pathway from the receptor all the way up to the specific spot in the brain it connects to is like a dedicated wire. A signal coming in on the optic nerve wire is always interpreted as light. A signal on the auditory nerve wire is always sound. That's the labeled line principle, reinforcing Miller's doctrine.
Got it. And what about telling exactly where a touch is or seeing fine detail? That's acuity, right?
Yes, discriminative ability or acuity. It's influenced heavily by the receptive field size, the area monitored by one sensory neuron. Smaller fields, like on your fingertips, mean higher acuity compared to, say, your back.
Makes sense. And you also mentioned lateral inhibition.
Crucial mechanism. The neuron that's most strongly stimulated actually sends inhibitory signals to its immediate neighbors.
So it quiets down the surrounding signals.
Exactly. It enhances the contrast between the strongly stimulated area and the less stimulated areas around it. It sharpens the perception. That's why you feel a single point when a pencil touches your skin, even though it's physically pressing on multiple receptors. The inhibition creates a sharper boundary in your perception.
Wow. Okay, so let's move into some specific senses. Mechanoreception sensing physical forces. And you said this is probably the oldest?
Very likely, yes. Even bacteria react to touch. We see it everywhere. Touch receptors in our skin, in insect exoskeletons. Often it's direct. Physical distortion literally pulls open an ion channel.
Like those TRP channels you mentioned earlier. In fruit flies.
Exactly, like the TRPN1 channel. And that TRT family is huge, involved in all sorts of senses across the animal kingdom, from yeast to us. It shows just how fundamental this mechanism is.
And proprioception sensing body position uses mechanoreceptors too.
Absolutely. Muscle spindles, Golgi tendon organs, they're constantly monitoring muscle stretch and tension. And yeah, back to fruit flies. If their TRPN1 genes are broken, they move in this really uncoordinated way. It's a very clear gene-to-function link for proprioception.
Okay, what about sensing orientation, like up versus down?
The simplest organ for that is the statocyst. Imagine a little fluid-filled sac lined with hair cells. Inside, there are dense particles called statolysts.
Like tiny weights.
Pretty much. Sometimes they're sand grains the animal incorporates. When the animal tilts or accelerates, the statolysts shift due to gravity or inertia, bending the hairs on the hair cells and triggering a signal. Super important for animals floating in water, like jellyfish or fish who don't have strong gravitational cues otherwise.
That makes sense. And that leads nicely into the lateral line system, in fact, doesn't it? That always seemed amazing.
It really is. It's a series of canals running along the fish's body containing these clusters of hair cells called neuromasts. They detect water movement currents, vibrations, pressure waves from other objects or fish.
So they can use it to avoid bumping into things, detect predators sneaking up, even stay perfectly positioned in a school.
All of that, yes. It's like a scent of distant touch through the water.
And the hair cells themselves. They're the key component again.
They are the transducers, yeah. And the basic mechanism is remarkably conserved. They have this bundle of hairs, stereocilia, arranged like a staircase. Tiny protein filaments called tip links connect the tip of one cilium to the side of the taller one next to it.
Okay.
When the bundle bends toward the tallest cilium, the tip links get stretched, and they literally pull open ion channels, cation channels. Calcium flows in for fish, potassium for mammals. That depolarizes the cell. Bend the other way, the tip links slacken, the channels close, and the cell hyperpolarizes.
It's like a tiny, incredibly sensitive mechanical switch.
Exactly. Elegant and effective.
And this same principle applies to our own balance system, the vestibular apparatus in the inner ear.
Yes, it uses very similar hair cells. Our vestibular system has two main parts. First, the semicircular canals. There are 3 of them, oriented in different planes. They detect rotational accelerations, spinning, nodding your head, turning corners.
How do they work?
Inside the canals is a fluid, endolymph. When your head rotates, the canals move, but the fluid lags behind slightly due to inertia. This lagging fluid pushes against a gelatinous structure called the cupula, bending the hair cells embedded within it.
Okay, so that's rotation. What about just knowing which way is up or moving forwards?
That's the job of the otolith organs, the uticle and the saccule. These contain hair cells covered by a gelatinous layer embedded with tiny, dense crystals of calcium carbonate the otoliths, or ear stones.
Ah, like the statoliths and the statocyst.
Very similar principle. Because the otolith layer is heavy, it shifts when you tilt your head or when you accelerate linearly, like starting to walk or jumping up. This shift bends the underlying hair cells, signaling your head's position relative to gravity and linear motion.
Fascinating. And the evolution of these is interesting too, like in whales.
Yeah, primates and birds that do complex 3D maneuvers have really large semicircular canals. But modern whales have tiny ones. Maybe it evolved to reduce sensitivity, prevent seasickness in their relatively stable aquatic world.
Maybe. And flatfish reorganizing their system after metamorphosis? Nature is just amazing.
It really is. The adaptability is incredible.
Okay, let's stick with mechanoreception, but turn to hearing. Perceiving sound energy. Also super important, especially for long-range warnings.
Absolutely. Sound travels through air, water, even solids. Fish, as we mentioned, use their lateral lines for low frequencies, and their otolith organs, those same balance organs, can detect higher frequency sound.
Really? The balance organs detect sound?
Yep. The vibrations travel through the body to the inner ear, and some fish have that Weberian apparatus, a chain of tiny bones connecting the swim bladder to the inner ear, which acts like an amplifier, boosting their hearing sensitivity significantly.
Clever. And on land. We evolved eardrums, right? The tympanic membrane.
Right. A vibrating membrane became the common solution. Though snakes do it differently, they pick up ground vibrations through their jawbones. Mammals, of course, have the outer ear, the pinna, to funnel sound down the ear canal to the eardrum.
And birds. They have amazing hearing, too. Barn owls.
Oh, barn owls are incredible. Their ear openings aren't symmetrical. One's higher than the other, and they face slightly different directions. This tiny difference in timing and intensity of sound arriving at each ear allows their brain to calculate the exact location of a sound source, like a mouse rustling, even in total darkness. It's like they're seeing with sound.
Wow. So sound itself? It has properties like liveness, pitch.
Right. Intensity is loudness, the amplitude of the sound wave measured in decibels. Pitch is the tone, the frequency of the waves measured in hertz. And timbre is the quality what makes a violin sound different from a flute playing the same note due to overtones.
And there are sounds outside our range, like infrasound.
Yeah, very low frequencies, travels huge distances. Elephants and whales use it to communicate over kilometers. Elephants might even sense it through their feet.
Amazing. And the opposite, ultrasound, high frequencies.
Right. Great for echolocation because high frequencies reflect well off small objects. Bats emit ultrasonic clicks and listen to the echoes. Dolphins do too, generating clicks with nasal sacs, focusing them with that fatty melon in their forehead, and receiving the echoes mainly through a fat-filled channel in their lower jaw that transmits sound to the inner ear. They can use the timing and timbre of echoes to see objects, maybe even tell what they're made of.
Incredible. So, okay, back to the mammal ear. Sound hits the eardrum. What happens next? In the middle ear.
The eardrum vibrates. These vibrations are passed along a chain of 3 tiny bones, the malleus and entius and stapes, or hammer, anvil, and stirrup. They act like a lever system.
Right, amplifying the vibration.
Exactly. They amplify the force about 20 times as they transmit the vibration from the large eardrum to the much smaller oval window, which is the entrance to the fluid-filled inner ear. This amplification is crucial to overcome the impedance mismatch. It's much harder to move fluid than air.
And those bones have an amazing evolutionary story too, right? From jaw bones.
Yeah, they're modified remnants of bones that formed the jaw joint in our reptile-like ancestors. A fantastic example of evolution repurposing structures.
Okay, so the stapes pushes on the oval window, sending pressure waves into the fluid of the inner ear inside the cochlea.
Correct. The cochlea is this coiled, snail-shaped tube. Inside, it's divided into fluid-filled compartments. The key structure is the organ of Corti, which sits on top of the flexible basilar membrane. This is where the actual hearing happens.
So the pressure waves in the fluid make the basilar membrane vibrate.
Yes, and as the basilar membrane vibrates, it pushes the hair cells of the organ of Corti up against the overlying tectorial membrane, which is relatively stationary.
And this bends the stereocilia, those little hairs on the inner hair cells.
Exactly. Bending opens mechanically gated ion channels. Potassium rushes in mammals. The cell depolarizes, releases neurotransmitters, and triggers action potentials in the auditory nerve fibers connected to it. That's the signal going to the brain.
But there are out hair cells, too. What do? They sound different.
They are fascinating. They don't primarily send sound information to the brain. Instead, they actively change length when their membrane potential changes. It's called electromotility.
They move.
Yes. They physically push and pull on the basilar membrane, amplifying its vibrations at specific locations. This sharpens the tuning, making the inner hair cells much more sensitive to specific pitches and intensities. They're like tiny amplifiers built right into the cochlea.
That's incredible. So how do we tell different pitches apart? High notes versus low notes?
That depends on where along the basilar membrane the vibration is strongest. The membrane is narrow and stiff near the oval window, and it vibrates most in response to high frequencies. It gets wider and more flexible towards the far end, which vibrates most in response to low frequencies.
So it's like a frequency map laid out along the cochlea.
Precisely, like unrolling a piano keyboard. And loudness. That corresponds to how much the membrane vibrates at that spot, the amplitude of vibration.
And this map is preserved in the brain, the ionotropic organization.
Exactly. Neurons originating from the high-frequency end of the cochlea project to one area of the auditory cortex, low-frequency neurons to another. And interestingly, signals from each ear go to both sides of the brain, providing some redundancy.
Makes sense. What about insects? How do they hear?
Their ears are often in weird places, like legs or the abdomen. They usually involve a tympanum, like an eardrum, often derived from their breathing tubes, the tracheal system. But their auditory systems seem more attuned to detecting patterns, duration, and loudness, rather than the fine pitch discrimination vertebrates are capable of.
Different needs, different solutions. Okay, let's switch senses completely. Chemoreception, taste, and smell. Super ancient, super important.
Absolutely. Finding food, avoiding poisons, recognizing mates or rivals, navigating. Chemicals are key information sources, and the sensitivity is just wildly variable.
Like cats not tasting sweets.
Right. Their sweet receptor gene is broken, a pseudogene. But then you have birds like petrels that can smell DMS, a chemical released when zooplankton eat phytoplankton from huge distances over the open ocean, helping them find food patches.
Wow. And pheromones, too.
Yeah, those chemical signals, often airborne, critical for social insects, and they influence mammal behavior, too. A male gypsy moth can supposedly detect a single molecule of the female's pheromone.
Incredible sensitivity. Okay, let's break down taste gustation. It happens in taste buds.
Mostly, yeah. On the tongue, in vertebrates, these buds contain receptor cells with little microvilli sticking out into the saliva, where dissolved chemicals, the tastants, can bind.
And binding causes a signal.
Yep. Binding changes ion flow across the receptor cell membrane, creating a depolarizing receptor potential, which triggers neurotransmitter release onto the connected neuron, sending the signal off to the brain.
And these taste cells get replaced constantly.
They do, about every 10 days or so. Unlike photoreceptors or hair cells, which generally have to last a lifetime.
So the primary tastes: salty, sour, sweet, bitter, umami.
That's the standard model. Salty is mainly detecting sodium ions, often via direct entry through specific sodium channels. Sour detects acidity, hydrogen ions, which can enter the cell or block potassium channels.
Cool.
Sweet and bitter and umami generally use G-protein coupled receptors. Binding triggers a cascade inside the cell, a second messenger system that ultimately leads to depolarization. Bitter involves a whole family of receptors, makes sense for detecting diverse toxins. Umami detects amino acids, that savory, meaty taste.
And gusducin, the G protein for bitter, is like transducin in vision. Cool connection.
Yeah, evolution reuses successful molecular tools, and now there's evidence for maybe a sixth taste for fatty acids, maybe even a seventh for calcium in some animals. It makes sense: fats, proteins, salts, acids, sugars, covering the major nutrient groups.
Plus, we can learn to like tastes, right? Like bitter coffee.
Absolutely. Perception isn't fixed. It's modulated by experience and context.
Okay, now smell, olfaction. How does that work? The receptors are in the nose.
In the olfactory mucosa, high up in the nasal cavity in vertebrates. And uniquely, the olfactory receptor cells are actually neurons themselves, afferent neurons whose endings are specialized to detect odorants. And they're renewable. They're the only mammalian neurons known to regularly divide and replace themselves.
Wow, replaceable neurons. So odorants have to reach these neurons.
Yes, they have to be volatile, airborne, and dissolve in the mucus layer covering the olfactory cilia where the receptor proteins are located.
We have fewer receptors than dogs, right?
Yeah, significantly fewer. Genomics shows there's a huge family of odor receptor or genes, hundreds or even thousands in some mammals. Humans have lost maybe 60% of ours became pseudogenes during evolution.
Linked to relying more on vision.
That's the prevailing theory, especially color vision in primates, a trade-off.
So how does the brain sort out all these smells from a limited number of receptor types?
It's fascinating. Each type of olfactory receptor neuron seems to respond strongly to only one or a few related chemical components of an odor. Then, all the neurons with the same receptor type converge on just one or two specific structures in the olfactory bulb called glomeruli.
So each glomerulus gets input about one specific molecular feature, like a smell file.
Exactly. You can think of it like that. The brain then recognizes a specific smell not by activating one rose receptor, but by the unique pattern of activity across many different glomeruli. It's combinatorial.
Like chords in music making a melody.
A great analogy. It allows us to distinguish thousands of smells using only hundreds of receptor types.
And smell adapts quickly too, right? That musty room smell fades.
Very quickly. Plus, there are enzymes in the mucus that break down odorant molecules, clearing the signal and maybe even detoxifying harmful inhaled chemicals.
And what about the vomeronasal organ, the VNO?
Jacobson's organ. It's a separate chemosensory system in many vertebrates, specialized for detecting pheromones, those social chemical signals. Often less volatile ones picked up by direct contact, like sniffing urine.
The Fleeman response in horses.
That's a classic example, drawing sense towards the VNO. It's strongly linked to reproductive and social behaviors. In mice, it detects proteins like darsin that signal male identity and status.
And the signal bypass the cortex, leading to subconscious reactions.
Often, yes. The pathways go more directly to areas involved in emotion and hormonal control, like the amygdala and hypothalamus. So it can generate these gut feelings, good chemistry or avoidance, without conscious processing. Some evidence even suggests it might detect signs of illness or tissue damage in others.
Primal stuff. Okay, let's move to photoreception. Vision, hugely important.
Dominant sense for many animals, over 90% of species have some way to detect light.
And the basic chemistry is ancient. Opsin and retinine.
Remarkably conserved. The basic photopigment machinery, using a protein opsin bound to a light-absorbing molecule, retinine derived from vitamin A, is found across the board, even in simple cnidarians like hydras. A deep, deep evolutionary connection.
And the organs themselves range from simple eye spots.
Right, like in flatworms, just telling light from dark, all the way to complex camera eyes like ours or in octopuses, and the compound eyes of insects and crustaceans. Incredible diversity.
And the PAX6 gene. That's the master control gene.
It seems to be. It kicks off eye development in everything from flies to mice to humans. And the really wild part is experiments where, say, a mouse PAX6 gene inserted into a fruit fly can trigger the formation of a normal fly eye.
It's mind-blowing. It suggests a single common origin for the intent to build an eye, even if the final structures are totally different.
Exactly. A master switch.
Okay, the vertebrate camera eye. Let's unpack that. 3 layers.
Yep. Outer tough layer is the sclera, the white, and the transparent cornea at the front where light enters. Middle layer is the choroid, pigmented, full of blood vessels, which forms the ciliary body for focusing, and the iris controls light entry at the front. Innermost layer is the light-sensitive retina.
And the iris works like a camera aperture?
Just like that. Circular muscles make the pupil smaller in bright light. Radial muscles pull it open in dim light. Controlled automatically.
And focusing is done by the cornea and the lens, bending the light.
Right. They both refract light because of their curved surfaces. The cornea does most of the initial bending, and the lens fine-tunes the focus onto the retina.
And the lens itself is made of amazing proteins, crystallins, evolved from enzymes.
Incredible story, isn't it? Duplicated enzyme genes repurposed for transparency and refraction, and many vertebrate lenses are actually multifocal, with varying refractive indices to correct for chromatic aberration, focusing all colors perfectly, something our simpler human lens doesn't quite manage.
And we adjust focus with accommodation, changing the lens shape.
In mammals, yes. The ciliary muscle contracts, relaxing tension on the suspensory ligaments, and the elastic lens bulges, becoming rounder and stronger for focusing on near objects. Relax the muscle, ligaments tighten, lens flattens for distance.
But fish move their lens back and forth.
Yep. And chameleons have muscles that squeeze the lens, actually changing its power significantly, even making it diverge light to magnify the image. Lots of different strategies.
Okay, the retina. Why is ours backwards? Light goes through other layers first.
Yeah, it's a quirk of vertebrate evolution. Light has to pass through layers of ganglion cells and bipolar cells before it hits the actual photoreceptors, rods and cones. It does create some issues.
Like the blind spot, where the optic nerve leaves.
Exactly. And it makes the retina prone to detachment. But nature compensates. Special glial cells, called Muller cells, act like fiber optic cables, funneling light through the intervening layers efficiently to the photoreceptors.
Clever workaround. And the fovea is where vision is sharpest.
Right. It's a small pit in the retina where the overlying neurons are pushed aside, so light has a direct path to a dense concentration of cones. That gives us maximum acuity. Dogs have a visual streak instead, better for scanning horizons. Some hunting birds have multiple foveas.
Multiple foveas, wow. And nocturnal animals have that reflective layer, the tapetum lucidum. Eye shine.
Yes, it bounces light back through the photoreceptors, giving them a second chance to capture photons, boosts night vision, and in some species, like certain lemurs, the tapetum contains riboflavin.
The vitamin.
Yeah, it absorbs blue light and actually fluoresces, re-emitting light at a wavelength the rods are more sensitive to, so it literally makes dim blue backgrounds appear brighter to them.
That is incredibly cool.
Yeah.
Okay, how does the light actually trigger a signal? Phototransduction.
It starts with the photopigments,
Yep.
opsin plus retinine, rhodopsin in rods for dim light, gray vision, different photopsins or scotopsins in cones for color. Now here's the weird part. In the dark, photoreceptors are actually depolarized.
They're on in the dark.
Yeah, sort of. High levels of a molecule called cGMP keep sodium channels open, so they're constantly releasing neurotransmitter glutamate, which is often inhibitory here.
Okay, so what happens when light hits?
Light hits the retinine, changes its shape, activating the opsin. This activates a G protein called transducin. Transducin then activates an enzyme that breaks down the cGMP. Less cGMP means the sodium channels close.
Ah, so the cell becomes hyperpolarized, more negative inside.
Exactly, it hyperpolarizes, and this reduces the amount of glutamate it releases.
So light inhibits the photoreceptor.
Correct. Light turns the photoreceptor off, or at least reduces its activity. This reduction in inhibitory glutamate release then excites the next cells in line, the bipolar cells, which then activate the ganglion cells, and they fire the action potentials that go to the brain.
That seems counterintuitive, but okay. So rods are for dim light, cones for bright light and color.
Right. Rods are super sensitive, lots of photopigment, lots of convergence, many rods feeding into one ganglion cell. Great for night vision, but not sharp detail or color. Cones need more light, provide sharp detail, less convergence, and give us color vision.
And the rod nuclei acting as lenses in nocturnal mammals. Nature finds a way.
Always.
So color vision depends on different types of cones, sensitive to different wavelengths.
Yep. Humans are typically trichromatic. We have cones most sensitive to long, reddy yellow, medium green, and short blue-violet wavelengths. The brain compares the signals from these different cone types to perceive the whole spectrum of colors. It's the ratio of stimulation that matters.
And other animals see differently.
Oh, hugely. Many fish, reptiles, birds see UV light. Birds can be tetrachromatic or even pentachromatic. Most mammals are dichromatic, like red-green colorblind humans. Primates re-evolve trichromacy, maybe for finding ripe fruit.
And the deep-sea dragonfish using bacterial chlorophyll.
Yeah.
That's just wild.
Yes.
Salmon switching cones.
Yeah, the diversity is stunning. And the mantis shrimp, up to 16 photoreceptor types. Mind-boggling what they might perceive.
We can barely imagine. But why did whales lose their blue cones? In a blue ocean.
Still a puzzle. Maybe their ancestors lived in murky coastal waters where blue light didn't penetrate well and they just never re-evolved it when they moved back to the open ocean.
Huh. Okay, so the retina processes the image. Upside down, lateral inhibition.
Right, sharpening edges. And you have these specialized ganglion cells, on-sender and off-sender, that respond best to spots of light or darkness with contrasting surrounds. They emphasize differences, defining contours.
And then the signals go to the brain in parallel pathways. For form, color, motion.
Yes, different aspects are processed simultaneously, and at the optic chiasm, information from the inner half of each retina crosses over.
So the left brain sees the right visual field and vice versa.
Exactly. And having input from both eyes for overlapping parts of the visual field binocular vision allows the brain to calculate depth based on the slight differences, the disparity between the two eyes' views.
And the cortex builds it all up hierarchically. Simple lines to complex shapes.
That's the model, yes. Increasingly complex feature detection as you move through different visual areas.
Yeah, cephalopod eyes. They evolved independently, and they're not backwards.
Right. Octopus and squid eyes have the photoreceptors on the top layer, facing the light with the neurons behind them. No blind spot, no backwards retina. A fantastic example of convergent evolution arriving at a similar camera eye solution via a different developmental path.
Amazing. And compound eyes in insects, little individual units.
Yep, the Ommatidia. Each has its own tiny lens and photoreceptor cells. It forms a mosaic image, generally lower resolution than a camera eye, but often a much wider field of view and excellent at detecting motion.
And their phototransduction is different too. They depolarize in light.
Yes, often using a different second messenger system. And that energy-saving trick in locusts, switching between day and night modes by regulating ion channels, potentially the fastest channel regulation known, just incredible efficiency.
Mind-boggling. Okay, quickly touching on a couple more senses. Thermoreception, feeling temperature.
Mammals have distinct warm and cold sensors, and again, TRP channels are key players. TRPV1, 2, 3 respond to increasing heat, TRPA1 and TRPM8 respond to cold. Some even react to chemicals. TRPV1 is the capsaicin receptor, chilly heat. TRPM8 responds to menthol, cool.
Direct link between the molecule and the sensation. And pit vipers. Infrared vision.
Essentially, yes. Those facial pits are incredibly sensitive thermoreceptors, detecting infrared radiation, heat from warm-blooded prey. They build a sort of thermal map of their surroundings.
Chilling. And nociception, pain. It's protective.
Primarily, yes. Alerting to tissue damage. You have mechanical, thermal, and polymodal, responding to multiple stimuli, including chemicals. Nociceptors, typically naked nerve endings.
And there's fast pain and slow pain.
Right. Fast pain is sharp, well-localized, carried by fast myelinated fibers. Slow pain is dull, aching, poorly localized, carried by slow, unmyelinated fibers, often triggered by chemicals released from damaged tissue, like bradykinin.
And TRP channels are involved here, too. TRPV1 for burning pain.
Yep, the chili pepper receptor again. TRPV2 for high heat fast pain, TRPA1 for cold pain, and also irritants like wasabi or chemicals from tissue damage.
And aspirin works because prostaglandins make these receptors more sensitive.
Correct. Aspirin blocks prostaglandin synthesis, and the brain has its own pain control, right? The analgesic system.
Yes. Brain regions like the periaqueductal gray can trigger the release of endogenous opiates, natural painkillers like enkephalins. These bind to opiate receptors on the pain pathway neurons, blocking the transmission of pain signals. That's why morphine works so well. It hijacks this natural system.
And that explains why birds eat chili peppers. They lack the receptor.
Exactly. Birds don't have the capsaicin-sensitive TRPV1 variant, so the peppers aren't painful. Good for the pepper plant birds to disperse the seeds effectively. Evolution is clever.
Always.
Okay, last couple. The really hidden senses. Electroreception.
Detecting electrical fields. Two types. Passive is just sensing fields generated by other things, like the faint bioelectric fields of prey. Sharks use their ampullae of Lorenzini for this, platypuses too.
And active, they generate their own field.
Right. Some freshwater fish have an electric organ, usually in their tail, made of modified muscle cells called electrocytes. They generate an electric field around themselves and then use other electroreceptors along their body to detect distortions in that field caused by objects or other fish, used for navigation, finding hidden prey, even communication.
Like the electric eel stunning prey.
That's an extreme example, yes, hundreds of volts. But many are weakly electric, like that knifefish, Sternopagus, that actively adjusts its signal strength between day and night to save energy by regulating ion channels, incredibly fast regulation.
Amazing. And finally, magnetoreception, an internal compass.
Seems so. Many animals, birds, sea turtles, salmon, even bacteria and mole rats navigate using Earth's magnetic field. How they do it is still debated.
What are the theories?
Three main ideas. 1. Magnetic induction via highly sensitive electroreceptors, like in sharks, detecting currents generated as they move through the field. 2. Magnetic minerals. Tiny crystals of magnetite found in cells, like in pigeon beaks, fish noses, might physically align with the field, perhaps pulling on ion channels.
Okay, and the third?
Magnetochemical reactions. The idea is that certain chemical reactions, possibly involving light-sensitive proteins called cryptochromes, are influenced by magnetic fields. Cryptochromes are found in bird retinas.
So birds might see the magnetic field.
That's the tantalizing hypothesis, like a magnetic compass overlay superimposed on their vision. Still needs more proof, but it's a fascinating possibility.
Absolutely mind-bending. Okay, so wrapping this all up, what's the big takeaway for someone listening?
Well, we've gone from these tiny ion channels all the way up to these incredibly complex and diverse ways animals sense their world. Whether it's a spider feeling vibrations on its web, a bat navigating by sound, or a bird using magnetic fields, it's just a symphony of senses out there.
Yeah, and so many are completely outside our own human experience.
Totally. And I think recognizing the limits of our own senses and how every species filters reality differently, it really promotes critical thinking, doesn't it? It makes you question your own assumptions about what reality even is.
Definitely. It opens up this huge space of possibilities for how life can experience the universe.
Exactly.
So maybe next time you're outside or even just watching your pet, take a second. Try to imagine seeing the world through echoes, or following an invisible map of smells, or feeling the lines of the Earth's magnetic field.
It really is humbling. Puts our own perception in perspective.
It does. Well, thank you for joining us on this deep dive. We hope this journey through animal sensory physiology has sparked some curiosity and maybe given you a new appreciation for the sheer sensory diversity of life on our planet.